This is a review of the specimens and the questions in Part B of the midterm exam. This is the first question that we had. Now, that doesn't necessarily mean it was the first question in your quiz, but I'm going to go through them systematically in the order that we originally made them up. And this question had to do with identifying the cell, the peripheral blood cell indicated by the pointer, and the only acceptable answer would be either a monocyte or monocytes. And the clues were there with the indented nucleus, the large cell, the largest of all these present, and the vacuoles in the cytoplasm. Those are all diagnostic characteristics, features of a monocyte. And if you zoomed in on it, you could see that even the one next to it is a neutrophil, and their neutrophil down here, there's a very large platelet. These are smaller platelets. If we zoom out, here's another neutrophil over here. And over here, this would be a large lymphocyte. And so some of the, actually 58% of the class answered correctly, and some of the incorrect answers were things like neutrophil, eosinophil, lymphocyte, and basophil, none of which were correct. So let's move on to the next question. This question indicated a lymphocyte in a peripheral blood sample, and you were asked in a white blood cell differential count on a normal blood sample, what is the highest percentage that would be represented by the cell indicated by the pointer? Well, the range that we gave you in the lecture was anywhere from 32 to 34%. So actually 33% is the exact number, but we allowed 32 and 34%. And initially, before regrading, 65% of the class chose the correct answer. And then when we regraded, we allowed the percent sign to be put in, so that just raised, and we also allowed 33% with percent spelled out. But that only raised the response correct rate to 66%. And we had numbers like 1, 2, 3, 5, 7, 8, 12, 15, 24, 30, 40, 45, 97. So it's clear that the class as a whole needs to review the percentage of the leukocytes in a differential white blood cell count. Let's move on to the next question. This specimen that was presented is actually a specimen of the heart, probably the left ventricle, where if you zoomed in on it, you would be able to see the features of cardiac muscle tissue. You can even see the turculated discs here, and you can see the nucleus in the center of the cell, and you can see branching cells. Here's a good example of a branching cell right here. And the question was, examine the tissue within the rectangular box, and this tissue forms what of the organ? And it's cardiac muscle tissue forms the myocardium of the heart. And the answers that we accepted were a variety of cardiac muscle, cardiac striated muscle, etc. We even regraded accepting striated cardiac and cardiac striated. So for the first blank, we did have some multiple answers, and after regrading, 85% of the class got that part correct. And then the others two, we found no alternative answers we could accept, and that would be 90% answered myocardium and 97% answered the heart or heart. And some folks put for the first part, put just muscle, or actually put myocardium and then middle in the heart. Some people put smooth muscle, etc. Some people put smooth muscle, tunica media, and the artery. And I would say in that case, it probably didn't zoom in to look and find, and identify this as cardiac muscle. Let's go to the next question. This question presented a specimen of bone, decalcified bone, stained with a trichrome stain. And you can see the calcified areas are red, and the less calcified areas still showing collagen blue, And then you can see the osteocytes in lacuna around here, and the center and the star. You were asked identify the round, almost empty space, contains a black star. 2 correct answers were accepted, Haversian canal, or we accepted Haversian's possessive canal. 85 or 88, 89% of the class got it correct. Some folks answered arteriole, another one answered central canal. I'm sorry, that's not specific. Others answered fibroblast, lacunae, lumen, macrophage, osteocyte, even sweat gland. So obviously, some of you really need to study bone and the features of bone. Next question. This question presented the epiphyseal plate and labeled the zones A, B, C, D, E. And you were asked in which location you would expect to find osteoid. Well, where you're going to find osteoid is where bone is deposited. And so if you examine these zones carefully and you looked at each one of them, you would see this is all cartilage. And you would see this is cartilage in which the cells are enlarging, the zone of hypertrophy. And then here you've got the calcified cartilage here. And then here is where you have bone deposition. And you can see that the osteoblasts are depositing pink matrix. And that pink matrix does not contain any of the osteocytes yet, and that is osteoid. So the only correct possible answer would be E. And we had answers, all of them, A, B, C, D, and E, but E is the correct answer. The next specimen. This question presented hyaline cartilage and encircled a group of cells. And these are chondrocytes that arose out of the mitosis of a original chondrocyte. And this is interstitial growth. And it's by a process of proliferation or mitosis, or we accepted a variety of answers. Initially, we accepted only interstitial or interstitial growth, and only 35% got it correct. But then we allowed, when we reviewed the answers typed in, we said, okay, we can allow proliferation, mitosis, and even interstitial growth of cartilage. So after regrading with these answers in the key, the percent correct rose to 72%. Okay, so let's go on to the next question. Now, this question is an example of one that if you failed to click on the annotation and then go to, you would not see exactly what we were asking about. So I'm going to click on go to, and you see that's what we're asking about right there. And it says the question, 2 profiles of a tissue are indicated within dotted lines. The tissue is composed of, and the answer we wanted was smooth muscle cells. And if you enlarge this, you could clearly see the tapered nuclei and cells, and you can see in the cross-section that some of the profiles of the smooth muscle cytoplasm have nuclei and the others do not. And this is, as we have tried to teach you, is that the nucleus is in the center of a tapered cell. So when you're doing cross-sections, if they're staggered like they are here, in some cases you're going to see the nucleus, in other cases not. Okay, so how did the class do on that? Well, the class did 70, 79, 80% correct. We accepted both smooth muscle, and we found that someone or a few put smooth muscles, so we accepted the plural of that. Go to the next question. Okay, this question presented 2 cells that were indicated by arrows, and the question said 2 cells are indicated by arrows. These are. Sorry, I had to pause there because my phone was ringing. I'm back now, and these cells are sensory ganglion cells. And if you enlarge them, you could see that the nucleus is here with a dark dot in the center. That's the nucleolus, and around it is the cytoplasm that is loaded with free ribosomes. Well, actually, no, little patches of rough ER, but not in a continuous network. This is very characteristic at the EM level of sensory ganglion cells. But that's not what the question was about. We just wanted you to identify them. And initially, only 21% got it correct. And I'll show you what we allowed in regrading. Initially, we accepted sensory ganglion neurons, sensory ganglion cells, etc. Here. And then upon looking at what you typed in, we found we could accept all these because we really didn't teach you the difference between sympathetic and sensory. If you mentioned ganglion cells or ganglion or anything that had ganglion in it related to cells, we gave you credit, and upon regrading, 62% of the class got it correct. So let's move on to the next question. This specimen is a blister. It's a blister of skin. And the question asks you, this is a specimen of, and the answer we wanted first was skin, and then the blank below, the dermis, is abnormally separated from the epidermis. And if you zoomed in on that, you could see this is dense irregular connective tissue down here in the dermis. And up here you could see that this was stratified squamous keratinized epithelium at this point. And then you have this big space full of fluid, which also has eosinophils and lymphocytes in it, and a lot of space with some protein exudate from the blood vessels, and a lot of tissue fluid forming the blister. Now, initially for the skin part, only 64% got it correct, but then we accepted a blister, and we also accepted integument as that first blank. And upon doing that, then the percent correct for the first part rose to 80%. The rest of them, there was no alternate answers we could accept. It had to be dermis or epidermis. And if I look and see what was answered, some even put liver or lung. We haven't even studied liver or lung. Some put lymph nodes. Those simply seem to be yes. Some people even put thymus, medulla, cortex, vein, tunica media. So I think that a lot of that is just not either not knowing it or not zooming in and looking at what you are presented with. Let's go to the next question. This question presented a longitudinal section of peripheral nerve and a cross-section of peripheral nerve, indicating certain structures. And we asked, the structure indicated in the center is in the center of what tissue component? So we're pointing to these dark dots here in the center of a nerve fiber, and surround that is the myelin sheath. And surrounding that between all these is the endoneurium. If we go back to the longitudinal section, you can see that dot now is shown in a line. And the actual precise name for that is the axis cylinder. It's actually in the center of a nerve fiber, and the light area is the myelin sheath. Now, we initially only accepted nerve fiber, singular plural, or nerve axon, singular plural. And when we did that, only 10% of the class got it correct. So upon looking at what others put in, what you put in, some said axon, the other said axons, and one person put myelin sheath. And so we accepted these a correct answer, and even myelin sheath is okay because the question asked, the structure is in the center of what? And truly it is in the center of the myelin sheath. So when we did that, then the percent correct in the class rose to 68%. But some people put nerve fiber bundle. That's not correct. Just neurons. That's not accurate enough. Just peripheral nerve. Remember, peripheral nerve is composed of fascicles of nerve, but we wanted peripheral nerve fiber, and some people put Schwann cell, spinal ganglion, epineurium, endoneurium, elastic fiber, collagen fiber. Just pay attention to the details. I'm going to go to the next question now. This question presented a specimen of a vessel, and it's on the edge of hyaline cartilage. Here's hyaline cartilage with the territorial matrix, the interterritorial matrix, the chondrocytes and lacunae, and the chondroblasts here forming new cartilage cells, and then the fibrous portion of the perichondrium here with a lot of collagen fibers. And out here you have some collagen fibers, you have lymphocytes, but here is this vessel, if you enlarge it, and you could have enlarged it more. Remember I told you you could press Z on your key and you could highlight this or use your marquee and see an enlargement. You can see this is just one layer of cells here, endothelium. And so that fits the description perfectly of a venule. We accepted venial, postcapillary venule. And of different, like you could separate post and capillary, put a hyphen between it, or run post-capillary together. We accepted venules in plural, so 86% of the class got this correct. And but some people put arterial, artery, capillary, collagenous fiber, etc., vein, transitional cells, pulmonary vessel, high endothelial venule. None of those were correct. Let's move to the next question. This question presented a specimen of bone. It's the cortical or compact bone. You can see the Haversian canals, some of them are large, where osteone is forming, and some are small that are completely formed. If you enlarge this, you could see the Haversian canal in the center. You see the outline of the concentric layers with lacunae and osteocytes. And you are asked to identify the dominant specific tissue type within the box. And we accepted compact bone tissue, Haversian bone, lamellar bone tissue, etc., etc. But some people put hyaline cartilage, others just put lamella. Others put osteocyte, some even put skeletal muscle or a thymocyte. So 75% of the class answered correctly on this one, and we didn't accept any alternative answers, except for all those that I just mentioned. This specimen was presented, and you were asked to identify the organ represented by the specimen. Well, first thing you should notice is you've got a lymphatic nodule with a germinal center. So then you need to ask yourself, what are the, and right away, you should suspect this is a lymphoid organ, right? And then you need to, what are the lymphoid organs that have lymphatic nodules? Tonsil, spleen, and lymph node. So what do we need to know to make sure this is a lymph node? Well, we can look at these nodules and see if we see an artery associated with it, and I don't see one, so there's no central arteries. What else could we look for? We can look for red and white pulp. I see only one shade. It's shades of purple or blue, and so none there. Of course, we do have a capsule, and under that capsule you have a gap with lymphocytes in it, so that should remind you of the subcapsular sinus, which is only present in the lymph node and not the spleen. So no question that it's not tonsil, because I'm looking around here, I don't see any epithelium associated with this. Over here you have a collagenous capsule. So the only possible answer would be lymph node. Yet only 59% of the class got it correct, and we had many answers said kidney, spleen, thymus, and tonsil. But the only correct answer would be lymph node. And a few people misspelled lymph node, and unfortunately we don't give credit for any misspelling. Let's move on to the next question. So here you were asked to identify the, based on the histology of the cells in this field, defined on the rectangular box, this specimen was taken from what organ? So you needed to zoom in and see the features of this tissue. And this tissue is cardiac muscle. While you cannot see the striations very well, because often in cardiac muscle, they don't show up that well. However, you can definitely see the nucleus in the center of the cell. It's kind of boxcar shaped. It's not long and tapered. You can see the branching here, and you can even see an intercalated disc right here. And I think perhaps there are others in the field, but certainly maybe one over here. There's an intercalated disc here. So the only correct answer is that this tissue is taken from the heart. So we accepted either heart or the heart. We found somebody put the heart down. But some people put medium vein, some people even put lumen, and another one put gastrointestinal tract. And I don't know why that was entered, because we haven't even studied the gastrointestinal tract. Others put skin, and some put medium vein, and even just plain muscle. So 96% of the class, however, got that correct. Let's move on to the next question. Okay, this specimen was presented. You were asked to identify the specific tissue that borders the empty space marked X within the box. So the first thing you had to do is make sure that you're looking at what we ask, and then to enlarge that and take a good look at the lining cells. And you can clearly see that they're columnar, and they're simple because there's only one layer, so it's simple columnar epithelium. And you were asked what tissue, so you've got to include epithelium in the answer. We accepted simple columnar epithelium. We accepted simple columnar epithelial tissue. And so since we actually mentioned tissue, then simple columnar epithelial tissue or just plain epithelium would be correct. But we had some folks who put stratified squamous, others put transitional, some would put tunica intima, some put integument, lymphocyte, connective tissue, adipose tissue, etc., etc. Some even put pseudostratified columnar epithelium, which they didn't recognize. It was only one layer of cells. And so 84% of the class, though, got it correct with the answers that we accepted. Okay, so let's move on to the next question. This question presented a specimen of bone, and you can see over here on the right, you can see where this was taken. This is a cross-section of a long bone, probably the femur, and you're looking at the very edge of it. So you got to think periosteum, and you got to think osteogenic cells, osteoblasts, things like that. And the question said, observe the cells between the two dotted lines, and these cells are. You're asked to identify these. And if you wanted, I mean, there's clues enough here because you have the bone here, and you have the fibrous portion of the periosteum here, and then these are osteoblasts. And so the answers we wanted were osteoblasts or osteoblasts. Now, in looking down through what people typed in, we saw that we could accept osteogenic, and looking right now at some of the it can't be the osteogenic cells, because these are already showing signs of osteoblasts. So we accept osteogenic because these are osteogenic. They're bone-forming cells, osteoblasts and osteoblasts. So with regrading, accepting in addition to osteogenic, we had 68% of the class got this correct. So we need to review that. Okay, this question presented a specimen of bone marrow, and a cell was indicated by a black arrow, and you were asked what it produced. So you can see the fat cells in here, and you can see the red portion of the marrow where you have the blood-forming cells. And this cell here you're pointing to is quite large, and it has a single nucleus. So that is a megakaryocyte, and it produces platelets. So there are only three plausible answers that we could accept: platelets, platelet, or thrombocytes, all of which are produced by the megakaryocyte, and 71% of the class got that correct. Now, some people put antibodies, other people identified the cell as a megakaryocyte, misreading the question, macrophages. Osteoclasts, remember osteoclasts have multiple nuclei, and they would be right on the edge of the bone in a Howship's lacuna where they're eating away the bone. Okay, so let's go to the next question. This question asks to identify, we're going to have to reduce this down, see the X, and you're asked to identify this vessel. A part of the wall is presented and has an X in its lumen. And so the first thing you're going to have to do is enlarge this and look inside. And the first thing you see are these elastic membranes. And there are multiple layers of these. And here would be the light staining endothelium. If I go to another area here, you can see the endothelium here. I'm going to move this down. And then if I move this up and we magnify this, so the three layers of this vessel are the endothelium and the connective tissue under it, which is the enema. And then the media is very thick, and then the adventitia out here. But your clue and the diagnostic feature were these elastic membranes. So the answers we accepted were aorta, elastic artery, or a large elastic artery. And 83% of the class answered correct. And we had some answers like medium artery, large vein, vacuole vein, vena cava, venule, or even adventitia, or even cardiac. So we're going to move on to the next question now. This question presented three profiles that were outlined with the dotted blue lines, and you're asked to identify the structure within that. And you look over here, and at the low power, you should recognize immediately this is a lymph node. You see the nodules in the cortex and the light center. That's a very typical pattern of a lymph node. And if you decrease the magnification, you can see the location of those. They are in the medulla, the lymph node, and they are the medullary cords. And so we accepted medullary cord or medullary cords, and we even accepted medulla cord. And 78% of the class with the regrading of medulla cord got that correct. But we had answers like lymphocyte, just plain lymph node, lymphatic vessels. Now the vessels, or the sinuses, are between these medullary cords. Some answered periarterial lymphatic sheath, platelets, sinusoid, thymus, skin, white pulp, all of which are incorrect. We even had one answer that was a versine canal. So some of you need to study that more careful on the lymphoid organs and their parts. This question presented two profiles of the same vessel, one stained with H&E above and the other stained with elastic stain. And right away you can see an elastic stain. You have a dark staining here and you have a dark staining here. And that suggests so what we needed to look at here is to identify, you're asked to identify the vessel. And so if you're looking at the H&E and enlarging it, and you look closely, you can see the media is composed of many layers of smooth muscle cells. And I don't know whether you recognize it or not, but this dark pink staining structure here is the internal elastic membrane. And if you zoom out, you can see the adventitia. But as soon as you get down here and look at the elastic stain, you can clearly see we have an internal elastic membrane and an external one out here. And then you have elastic fibers out in here. And you have a few elastic fibers in here, but no membranes. The rest of it's composed of smooth muscle, whereas all this pink areas here. So that is a medium-sized artery, muscular artery. We accepted muscular, distributing, medium-sized artery, and 68% of the class got that correct. Now, some people said it was a large artery. That's not correct. Others just answered pulmonary. Why you would put pulmonary when we haven't even studied pulmonary artery yet, I don't know. And then a tunica and vein and vena cava, arteriole, aorta, just plain blood vessels, not specific enough. So let's move on to the next question. This question simply asked, identify the structure enclosed within the dotted line. Well, the answer that we wanted was central artery, or we even accepted artery in this case. But if you zoomed out, you could see that you have collection of lymphocytes on the side of this, which suggests a nodule, and then you have a similar vessel right in the middle of surrounding lymphocytes, which you could imagine that that's the periarterial lymphatic sheath. And then if you look at low power, you can see red and blue. So without question, this is the spleen. So it has to be the central artery. And 63% of the class said either central artery or artery. But we had answers like Hassel's corpuscle, blastocyst, blood vessel, nucleus, large artery, sinusoid, etc., etc. We did accept arteriole, and so that's okay. So anyway, let's move to the next question. I won't spend a lot of time on this one because the 92% of the class got it correct. Let's identify the layer of this organ located between the two dotted lines. And this actually was a medium-sized artery, a muscular artery. You can see the smooth muscle cells here, internal elastic membrane, an external elastic membrane here, and this is the Adventitia. Oh, sorry, I hit the wrong. Okay, so this is the Adventitia, and we accepted any variation of tunica adventitia, as you can see here, even tunica externa, we gave credit for that. But we didn't give credit for things like perineurium, intima, integument, endothelium, basal lamina, tunica media, transitional epithelium, etc. So anyway, let's move on to the next question. This question, I'm going to click here and go to, and that brings right up what we're asking about. What specific name is given to the round cells with blue staining heterochromatic nuclei in the region enclosed within a yellow dotted line? Well, again, you got to recognize these are lymphocytes. If you've got such blue dense staining, and particularly if you zoom in, you're going to see, yes, they're lymphocytes, but we want the specific name. So we initially said any variation of T cells, T lymphocytes, T cell, whatever, singular, plural. And then after looking through all of the answers, we accepted mature T lymphocyte, singular, plural, naive T lymphocyte, singular, plural, naive T cell, and T lymphocyte. But even so, we only increased percent correct from 35.6 to 36%. So you might wonder, what are all the incorrect answers that were put in? Well, we had things like dendritic cell, we had Hassel's corpuscle, epithelioid cells, epithelial reticular cells, we had isogenous group, we had just plain lymphocyte, that's not specific enough. We had splenic nodule, serous cells, even thymocytes. Now, you see thymocytes, that's the name of all the cells here, but you have to be specific. These are mature T cells by this time because they're going through a process of selection in the cortex. Just review your lecture material on this. We'll go to the next slide. This question asks to identify the tissue throughout the specimen stain intensity is in, the tissue enclosed, then the dotted line is typical, and it was dense irregular connective tissue, and certainly looking at this high magnification, you see the fibers are not parallel, and the fibroblasts on the outside of the collagen fibers are clearly here with the elongated nuclei, and 88% of the class got it correct. But we had some people that put fibrocartilage, endoneurium, stratified epithelium, loose connective tissue, just plain collagen or collagen fibers, or just plain connective tissue. None of that was correct. Okay, let's move to the next slide. Okay, this question asks you to identify the specimen. So this is a specimen of skin. And actually, we accepted skin or thin skin, and we also accepted integument. And you could, if you zoomed in, you could see a stratified squamous keratinized epithelium at the surface. And then zooming out, you can see the rather loose connective tissue with very fine collagenous fibers in the cells very far apart, fibroblasts, nuclei all over the place. Then as you go into the dermal, from the papillary layer of the dermis to the reticular layer, you see the thick collagen fibers. And then down here you have some smooth muscle cells, a part of the erector papillae of hair papilla. So 84% of the class chose either skin, thin skin, or integument. We had some people answer bladder, just plain epidermis, esophagus, gallbladder, tonsil, vagina, urinary bladder, lung. Obviously, many of those were guesses. Okay, so let's move on to the next question. Now, this question, we ask you to identify the type of tissue in this structure, in these three structures. Well, actually, what we wanted was peripheral nerve tissue within these structures, and an expert histologist would recognize that this is nerve tissue just from the pattern of the cells located in here. But we concluded eventually that was unfair, because you did not see any clear longitudinal or cross section of the nerve fibers. And so we gave everybody credit for this question. If we ever ask this again, we'll make sure you have the right clues. So everybody got credit for that question. This question presented epidermis that's keratinized, and it pointed to these 4 cells. And these cells are Langerhans cells. You see they have an irregular shaped heterochromatic nucleus with a light unstained cytoplasm surrounding them. And they're in the stratum spinosum, very characteristic of Langerhans cells. And the question said, what organ does this cell often end up relocating as a part of its function? Remember, Langerhans cells capture antigen in the epidermis, and then they migrate down in the tissues, get into a lymphatic capillary, and eventually end up in lymph nodes where they present the antigen to T or B lymphocytes. So only 56% of the class chose lymph node, lymph nodes, or lymph node that we gave credit for. Other answers that were put in were axon, bone, central artery, dermis, keratinocyte, kidney, matrix, skin, spleen, stratum corneum, and even thymus. So need to review Langerhans cells and the relationship to antigen capturing, presenting, and lymph nodes. This question presented this specimen of tonsil. And this is actually the lingual tonsil, because it has associated with it stratified squamous non-keratinized epithelium you can clearly see here. And this is very typical of the oral mucosa or the esophageal mucosa, because it has a lot of lightly stained cytoplasm, which it really is glycogen. But the clues for tonsil are that, as well as the nodules here lining the crypts. Now, we accepted palatine tonsil, just plain tonsil, or tonsils, lingual tonsil, and we even accepted pharyngeal tonsil, because if you recognize this tonsil, that would be just fine. But some people thought it was rectum. Why, I don't know. Spleen, thymus, and even some people put lymph node and also liver. Let's go to the next question. This question presented a nodule in the spleen. Identify the structure enclosed by yellow dotted line. But you did have to recognize this is spleen, so it's a good idea to go at lower mag and see the pattern of blue and red, suggesting white and red pulp, and then enlarging this and notice the artery to one side, the central artery, and notice that the nodule does go to one side. That's why it's called a central artery, but it's really on one side of the nodule. And some people put periarterial lymphoid sheath, but that's only around the central artery. And when there's not a nodule, that continues. Like over here, this would be the periarterial lymphatic sheath around this vessel. But we ask about this over here, which was a nodule, and you could have answered splenic nodule, splenic lymphatic nodule. You could answer with the plural. You could even say white pulp primary nodule would get credit. But you couldn't say periarterial lymphatic sheath, which some people put, like red pulp, just plain white pulp. We asked specific name, trabecula. Hassel's corpuscle was entered. That's incorrect. Central artery and just even plain blood vessel was entered, but that was incorrect. But you know, many people in the class missed this. It's only 10% of the class got it correct. But we just have to stick because the only correct answer is the ones that I gave to you. Now we're going to look at the last question. This question presented this specimen and it said identify the organ. And so the organ actually is the heart. And here's the epicardium, the myocardium, and endocardium here. But for you to be sure about it, you would need to increase the magnification and look at the branching cells, the nuclei in the middle of the cell, and then look around at maybe some of the other areas where you should see greater profiles, better profiles. The cross section, if it was smooth muscle, they'd all be a uniform round, small, medium, and large. But here you have irregular profiles suggesting branching, like here. So after examining different areas, you should have concluded that this is the heart. And we gave credit for heart, and we also gave credit if you typed in the heart. And 91% got it correct. But we have people putting things like cardiac muscle, which we didn't ask that. Interventricular septum, we didn't ask that. Liver, lymph node, pancreas, thymus, and tongue. Okay, so that concludes. This is a little more than 42 minutes long, but it takes a while to go through all of these, and I trust this will be helpful as you study the remaining part of the course and the remaining organs and prepare for the final exam. Good luck.